Your genes are basically expressed as a polycistronic mRNA. So basically during the transcription part, the DNA is being transcribed into polycistronic mRNA. Polycistronic mRNA means that um, they have multiple, um, so there are the multiple genes are encoded into one mRNA. Within one mRNA sequence, it encodes for multiple genes, right? And so yeah, so that's what we call polycistronic mRNA. And so and also there's no like post transcriptional modifications in prokaryotes. There's no alternative splicing in prokaryotes and stuff like that. And so with regards to eukaryotes though, they do have the membrane bound organelles, they do have the nucleus. They also usually on one mRNA encodes for only one gene product, and also they have alternative splicing. So alternative splicing means that there can be multiple possible combinations of the exons with each other, and these are basically regulated by the introns actually, because the introns are basically binding sites for proteins that facilitate the splicing itself. Sometimes the mRNA can splice by itself, and we call them ribozymes. But yeah, and also remember how the prokaryotes have this plasmid, which is the small circular genetic material, small circular DNA that is basically separate from the genomic DNA, and its functions as a it's for horizontal gene transfer, right? And so basically, remember how I talked about the prokaryotes having the polycistronic mRNA, and so because multiple genes are basically encoded within that mRNA, and multiple genes are usually are side by side in the DNA itself. So they have what we call an operon. So operon is basically a functional unit within the DNA in which multiple genes are controlled or regulated by a single promoter, which allows for coordination of these multiple genes that function similarly. For example, the lag operon. So I talked about the lag operon last time, right? And so the lag operon itself in here, where's lag operon? So lag operon is remember is multiple genes that is controlled by one promoter, and in the case of the lag operon, those genes encodes for the enzyme that can break down and metabolize lactose. So those enzymes would be like Z, like A, like Y. So yep, so those are enzymes that are responsible for metabolizing lactose. And so they're controlled by one promoter, and this is what we call a lag operand. So the lag operand, within the lag operand itself, there's the operator. Operator is basically binding site for the repressor. There's a promoter binding site for the RNAP, and then there's the repressor gene. Oh yeah, repressor gene. So in the case of the repressor gene, is basically the one that will bind to the operator. So when there is no lactose present, what happens is that the repressor will bind to the operator, right? Repressor binds to the operator, and also the cap protein, this cyclic AMP cap protein. The cap protein will bind to its cap binding site. So if I just draw it in here, you can see that in here there's the cap binding site. And the cap binding site, there's the promoter binding site, there's the operator, and this is the lac Z, lac A, and lac Y. Lac A, lac Y, lac Z, and these are the genes for lactose, for metabolizing lactose, right? Oh no, upstream to this gene, there's a gene that encodes for the repressor. So basically what happened is that when there's no lactose present, the repressor binds to the operator, and the cyclic AMP cap also binds to its cap binding site, and there's no repression because even though this promoter might bind to the, so in order for a promoter for the RNAP to bind to the promoter, you need the cyclic AMP cap to bind to it. So cap binding is required for the binding of the RNAP to the promoter. And so although the promoter can bind, and because of the presence of the repressor binding on the operator, there's no gene expression. So there's no expression of genes that's important for breaking down of the lactose, right? When there is lactose present, however, the allolactose will bind to the repressor, which will then prevent the binding of the repressor on the operator, and then the binding of the cyclic AMP mediates the binding of the RNAP towards the promoter, and gene expression can occur, in which all of these LAC-Z, LAC-Y, LAC-A are expressed to basically metabolize the what was the name of that thing? Metabolize the lactose, right? Yeah. But then when there is both glucose and lactose, although the allolactose will bind to the repressor, and which prevents the repressor binding to the operator, because there's glucose present, glucose will bind to the cyclic AMP cap protein, which prevents it from binding to the cap binding site, which prevents the binding of the RNAP to the promoter, which means that there's no expression of the genes for metabolizing lactose, because they would prefer to actually use the gene to break down glucose first, because glucose metabolism is kind of like the main pathway, the main way we get energy. Our primary source of energy is basically metabolizing glucose, and so those genes are usually already active, and so instead of activating more genes, expressing more genes, which requires energy for metabolizing lactose, you might as well just use glucose first, right? So that's kind of like the rationale behind it. And so if you think of it like this, so when there is both glucose, right, and then there's lactose, and when there is glucose and lactose, so lac operon activation can only occur when there is no lactose and there is glucose. Lac operon on. So there's no glucose, but then there is lactose, right? If there is glucose and lactose, the genetic circuit looks like this, then lac operon off. If there's glucose and no lactose, lac operon also off, because there's no lactose. Yep. When there's glucose and the lactose operon is off. And so yeah, so that's what happened. So just remember that the glucose will basically bind to the AMP cap protein, which prevents its binding to the cap binding site, which prevents the binding of the RNA to the promoter, which means no activation of the lac operon. And another type of the operon, it's the I believe it's the tyrosine one. Was it the tyrosine operon? If I'm not mistaken. Polycistronic mRNA. Yeah, so polycistronic mRNA is basically a mRNA sequence that contains that encodes for several genes. Central, yeah, there's also central dogma. So, for example, here, in sickle cell anemia, describes the mutation responsible for the disease. So basically, in sickle cell anemia, there's a mutation in the HBB gene. So the HBB gene basically encodes for the the beta globin of the hemoglobin, and there's a substitution. So if it's a point mutation, it's a point mutation which leads to a change of the amino acid, so that would be a missense mutation. A nonsense mutation would be a mutation which I believe it creates a non-functional protein. That's a nonsense mutation, right? There's a frame shift mutation, there's a nonsense mutation, there's a missense mutation. I already forgot all of these, to be honest. Wait, let me check. A nonsense mutation. Mutation. Yeah, intrusive prematures. Oh, nonsense mutation enters a premature stop codon in the gene, which then leads to a non-functional protein. Either it terminates the translation early, or which occurs a non-functional protein by introducing stop codon, and then a missense mutation. A single nucleotide change results in a right and then sickle cell anemia mutation. A point mutation, yeah, on chromosome 11. Wow, HBB gene on chromosome. So remember, a point mutation which results in the change of the amino acid sequence is called the what do you call that? It's called the missense mutation. So because of a single nucleotide change that results in the different type of codon that is produced, which result in different amino acids. So that was from glutamate into valine, and so that was a missense mutation on chromosome 11. So HBB gene. So what is the HBB gene? I believe HBB gene is the one that encodes for the beta globin, if I'm not mistaken. Yeah, the HBB gene encodes for the beta globin and the hemoglobin. I knew it. Yeah. Hi. I think I'm sick. So just remember that there's a mutation, a point mutation, specifically a missense mutation in the HBB gene, chromosome 11, which changed the glutamic acid into valine. And so because of that, there's a basically then leads to a misfolding of the protein. So not misfolding, I guess a misfolded protein, like a mutant protein. The conformation is not right of the protein, and this is because in the T state, when there's low oxygen level and it's kind of like a deoxy hemoglobin, the glutamic acid residue, the glutamate residue, is actually exposed, which is fine when you have the glutamic acid, because the hydrophilic one will just solvate, the water will just solvate the glutamate acid residue. But when you have the valine is quite hydrophobic, and so exposing that into the aqueous environment will then lead to the hydrophobic effect, which means that because there will be the water cannot solvate the valine residue, there will be formation of the ordered cage structure around the valine residue, which decreases the entropy. And to mitigate the decrease in entropy, they would want to decrease the surface area by aggregating together. And so the aggregation of these multiple sickle-celled proteins, the hemoglobin, creates kind of like a clot, can create blood clot, and they cannot basically transport the oxygen. Yeah. And so what is the mutation in the DNA sequence of the HBB gene? And so how do they basically cure this disease using CRISPR-Cas9? They don't edit the HBB gene, they edit the BCL11A gene. BCL11A gene encodes for a encodes for a repressor for the fetal hemoglobin, HbF. And so by basically knocking out the gene that encodes for repressor, you can activate the fetal hemoglobin, and fetal hemoglobin will replace the adult hemoglobin. Describe the downstream consequences at the transcription and translational level. I already know that. Hypothesis as to why, I already know that. And gene more express. I didn't know this. I mean, a gene, remember, a gene is basically a functional region within the chromosome that encodes for certain gene products like proteins or RNA or other stuff. And so the coding sequence within the gene itself is a sequence of the DNA that directly encodes for a certain gene product, right? Directly transcribed to mRNA. That encodes for a gene product, which means it's being transcribed to mRNA and being translated to protein if the gene product is a protein, or sometimes it's just translated to a type of RNA like a tRNA, mRNA, sometimes microRNA. So it's not always proteins. Regulatory elements within the coding region, within the gene, is basically DNA sequences that is the binding site of regulatory proteins that control and regulate the gene expression itself. So, for example, it can be the binding sites for the promoters, for the RNAP, which is called the promoter. For example, in the binding sites for repressor in bacteria is called the operator. In human and eukaryotes called the silencers, and activators, enhancers. Remember, silencers are the binding site for repressors. Enhancers is the binding site for activators. So things like that. And non-coding regions. So non-coding regions within the DNA sequence are basically what we call the introns. So introns are non-coding regions, which will get spliced out during the post-transcriptional modification using alternative splicing. Usually introns are still important because they're basically the binding sites for certain proteins that mediate splicing itself. So basically, depending on what type of introns, you can have alternative splicing. Yep. Function of gene, protein coding genes, basically genes that encodes for certain proteins, basically kind of like the manual for making proteins. Types of RNA, there's the tRNA, there's rRNA, and there's mRNA. Mature RNA is basically the one that will be encode for proteins. Well, tRNA is basically important in in the translation. So tRNA, amino acid conjugate, the tRNA will be conjugated to amino acid because they have the ester bond, and the ester bond is higher in energy compared to mi bond because remember, the ester bond is not as efficient in electron delocalization, which means it's higher in energy. The carbonyl bond is not as stabilized. The carbonyl carbon is not as stabilized. There's a larger Del plus, and so basically tRNA is the one that will be conjugated to the amino acid, which will then be delivered into the ribosome for translation, right? And then for the rRNA basically are the ones that make the ribosomes. Yes, the ribosomes and the tRNA. Remember in here you have the tRNA also. An important thing about the tRNA, they have this like the hairpin type of structure. So they have a lot of the palindromic sequence. So the palindromic sequence, remember, it can be important for recognition, for selectivity. For example, in the restriction enzyme, they recognize certain palindromic sequences at DNA level within the DNA sequence that leads to the cutting of the DNA, the cutting of the double-stranded DNA by breaking the phosphodiester bond. Sometimes palindromic sequence is important for structural parts. So for example, in the tRNA, there's a lot of the palindromic sequence that makes a kind of like a hairpin structure that basically makes the tRNA secondary structure. And remember, the tRNA has this the hydroxy group that will be attached to the amino acid. And then mRNA processing, remember, it's pretty easy. I mean, there's the splicing, there's also the poly-A tail attachment, and then there's also the 5' G cap, 5' capping, poly-A tail thing, and make sure so basically all of these mRNA processing from the Alternative splicing. Alternative splicing is basically to have a variety of the gene product. So depending on the splicing, you have different type of the proteins that is translated from the different type of the alternative splicing. And for the polyatase to basically make the RNA more stable from degradation. 5' capping is also important for basically preventing degradation, and also it facilitates export of the mRNA from the nucleus into the cytoplasm for translation. Yes. Yeah. There's a lot of types of RNA: microRNA, silencer RNA, RNAi, RNA interference. Define genetic engineering. So genetic engineering is basically the I guess the manipulation of genetic materials by means of, let's say, DNA recombinant DNA or DNA or modification within the genetic material itself. So modifying either DNA or RNA to make useful product. This is an important J. Muller's experiment on the fruit fly, in which he exposed X-rays to induce mutations. Huh? J. Muller's? Who the fuck is J. Muller's? The fuck? Oh my god, he wants us to freaking remember the history. Oh my god, I hate him. I hate this professor. I hate mosquitoes. Where is that stupid J. Muller's? J. Muller's? Who is J. Muller's? Is this J. Muller's? Bro, who is J. Muller's? Let me just type it. J. Muller. Huh? Who is this? Fruit fly experiment. J. Muller fruit fly experiments. Bro, who is he? Where? He didn't even talk about freaking J. Muller's. Who the fuck is J. Muller's? I hate him. I hate him. Explain the 3 major techniques for genetic engineering. First, there's the DNA recombinant DNA, in which you basically use what was the name? Recession enzyme and DNA ligase to genetically modify things. There's a CRISPR-Cas9 type of gene editing to basically introduce knockout mutations or certain mutations. Other types of genetic engineering. I mean, you can also say like talons or zinc fingers. Did he even mention three major techniques? He didn't even mention like the major techniques. What's a major technique? How do you define the major technique? Is this what he was referring to when he says major techniques? Recombination, nuclear transfer, cell fusion. Is that the 3 major techniques? Is that really major? How do you define major? Ugh, annoying guy. So annoying. I really hate him. Ugh. I hate my professor. Anyway. Oh well. But he didn't even explain nuclear transfer or cell fusion. I mean, I know what nuclear transfer is. You're just transferring all of the one nucleus from one cell into the other. Cell fusion. Oh, cell fusion. I learned about this because in cell fusion, you basically fuse 2 cells together, and so if the 2 cells are in different stage of the cell cycle, the basically the proteins, the RNAs, and all of the molecules in the cytoplasm of that one cell can be transferred to the other cell, and that will induce, or that will change the cycle of the other cells. For example, if the one cell is in, let's say, a certain, maybe in the M1 phase, M1, is that M1? G1, let's say in the G1 phase of the cell cycle. And then this guy is not in the G1, it might induce it into go to G1, or it might induce it into go to the mitotic phase, so the M phase, maybe. So yeah, so that, I think that's what he was referring about, the cell fusion. Cell fusion, nuclear transfer, DNA recombination. Okay, but this is stupid. Ugh, stupid shit. I hate this class. Ugh. Explain where genetic engineering belongs in the spectrum of biotechnology. What kind of stupid question is this? Explain where it belongs in the spectrum of biotechnology. Why? Why do you need to define a spectrum of biotechnology? What's the use of this shit? Ugh. Explain the significance of experiments performed by... Why do we need to know certain... What was the first recombinant medicine in history? Insulin, probably. List the fields where genetic engineering is applied: agricultural, medicinal field, sustainability, food. What are examples of genetic engineering application in the field of pharmaceuticals? Making insulin, what else? Creating artemisinic acid, which is a precursor for artemisinin. What are examples of substances producing animals as a bioreactor? Insulin. Another example are artemisinic animals, and no, not animals then. Maybe it was goat, or something. Was it pig? Yeah, I mean they used pig, yeah. And so I can say insulin probably, or certain metabolites. I don't remember. Oh, antibodies. Yeah, anti-snake venom. It's used from antibodies of horse. Give some examples of the application of genetic engineering. Field of food engineering. BT corn. There's some stupid shit. Ugh, I hate this shit. Understanding genetic engineering. Why? This is some stupid shit. I hate this shit. Misformed protein. What the Who in the... What? Various inorganic substances, and you mentioned methane. Who wrote this? Why is he using... Ugh, my god. Ugh. Incompetent professor. Including radiation from sun, phosphoric acids, which is assumed to originate from rocks erupted by volcanoes without the formation of RNA polymers. The RNA world hypothesis is where RNA is like the first kind of like a biomolecule that exists before DNA and proteins. So RNA can, they're basically arguing that RNA can self-assemble, and also they can basically produce protein through the help of, they can produce proteins because ribosomes are made out of RNA. And also, from then one of the proteins that are formed, one of them would be reverse transcriptase, which would explain the formation of the DNA. Yeah, Mr. RNA carries genetic instruction to represent transfer RNA, tRNA. Yeah, bring the RNA to the ribosome. So ribozymes are RNA that can act as enzymes. For example, in the what do you call that? Some RNA can act as a spliceosomes. Basically, they can splice their own like exons and introns. Yeah, so RNA enzymes, that would be a type of ribosomes probably. Yeah. Ribosomes are RNA enzymes. Yeah, from AT to TA. It's a missense mutation. It's a point mutation that results in the codon change, which results in the different amino acid that is incorporated within the protein. Yeah, so this glutamine is valine. A to T, glue to valine, which means the mRNA would be T into U. Wait, what? Sorry. So this is a template DNA because it's 5' to 3', and so it would be A, so it would be T into A, right, in the DNA level. In the RNA level, it would be A into U, right, and then it would be a glutamate into valine. Much. So tomorrow will be my last exam. Okay? Was it lagging? I think it was lagging. Maybe it was lagging. Is this love? I hate my professor. And epigenetic regulations, type of epigenetic regulation is basically how accessible the DNA sequence is for transcription or for DNA replication, for example. And so those are mediated by methylations, for example, and also acetylations. So those are a type of acetylation, methylations. Methylation would be on the DNA sequence. But for histone modification, you can methylate histones, you can also acetylate histones. So remember that histones itself, they have a lot of the positively charged lysine and, was it arginine? I forgot. No. Arginine is, I forgot. Histones are a positive charge amino acid residue on histone. I know that there's lysine, I just forgot there's another one. Lysine and arginine, yeah. So there's lysine and arginine, right? And so because of that, because of these positively charged lysine and arginine residues, they're able to bind into the negatively charged phosphate backbones. And so that's how DNA gets wrapped around histones. And so by acetylating the lysine, because remember there's an amine group, you're putting the acetyl group on the amine group, so you're basically neutralizing the charge, and so it will not be able to bind very well to the DNA phosphate, a negative charge on the DNA, which means that it would be more loose. And so if you have a very like a loose chromatin, it would be euchromatin, while a more tightly bound chromatin would be heterochromatin, right? And so yeah, and also DNA methylation is usually DNA methylation, they recruits other proteins that lead to the formation of the heterochromatin. So DNA methylation usually results in more tightly wound DNA, like less accessible DNA, yeah, more compressed DNA, I would say. Transcription regulation, transcription factors, promoters, enhancers, silencers. Processing phosphorylation. Glycosylation. Remember, operands are basically functional units. Functional units within the DNA where basically multiple genes are regulated by one promoter, which allows for the coordinated regulations of genes that have similar functions. For example, in the lac operon, genes that are used to metabolize lactose are controlled by the one promoter in the lac operon. And then there's another type of operon, which is a tryptophan. Okay, tryptophan, not tyrosine. I always forget. So tryptophan is a type of aromatic amino acid, and so all of the components to make the tryptophan is encoded within the genes, right? And then within those sequence of genes that are controlled by one promoter. So there's a tryptophan, the TRP operon. So with regards to TRP operon, again, the tryptophan is a co-repressor. So when there's no tryptophan, the repressor will not bind to the operator, which means that you need to synthesize more tryptophan. But when there's enough tryptophan will bind as a co-repressor, and then to the inactive repressor, and then do conformational change, leads to the binding to the operator, which means that it will there's not going to be a gene expression of the TRP operon. Yeah. And then so then that means that when there is no TRP TRP off, TRP operon, when the TRP operon is on, when there is no tryptophan. When there is tryptophan, then it will be off. TRP operon off. And then molecular cloning, blah 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 blah. I already know this. I'm speechless. Why are you speechless, bro? Plasmid also divide independently, express genes, and recently manipulated. So remember that DNA absorbs UV. Is it UV? Yep, I think so. It's UV. This DNA absorbs UV at 260 nanometer, while RNA absorbs UV at 280 nanometer. While other stuff, solvents and salt contaminants absorb UV at 230. So if you look at this, you can see that there's 100% DNA when you absorb it at 260, and this is the absorption at 230, which means it's solvent, right? And then for absorbance at 280, that means there's a protein contaminants. And for RNA, RNA usually is also the same. It absorbs at 260, but RNA will have a higher absorbance. Yeah, more absorbance of RNA compared to double. Well, single-stranded RNA will have more absorbance than higher absorbance than the double-stranded DNA, because the pi stacking of the double-stranded DNA kind of like it's not as effective in absorbing the UV light. UV this. Yeah, the RNA instability is because of the hydrolysis from the two prime, basically internal nucleophilic attack of the phosphodiester bond by the 2' hydroxy group. And then RNAs. And then DNA denaturing. So denaturation step is usually happens at 96 degrees. Okay, that's denaturation. And then renaturation is like cooling it down. So polymerization reaction. Oh my god, did you guys know? So do you guys know polymerase chain reaction, right? So there's this DNA molecular technique in biology called PCR. The guy who invented PCR was high on acid when he invented it. So he was driving. he was driving. I think he's from California or something like that. He was driving, and he was taking acids, which is like the drugs. So he was taking drugs, and then suddenly he was like, "Oh my god, I got this brilliant idea to basically synthesize DNA," and he came up with the polymerase chain reaction. That is crazy. So maybe the key into becoming like geniuses is taking drugs. I'm kidding. The forward primer, there's the reverse primer. What's the correlation between your term and that man? Who is that man? What do you mean, that man? Who is that man? Any enigma. What is enigma? What is enigma, huh? The one, the PCR one. No, it's just a background story when we were learning PCR. Got into acid. What is enigma? I don't understand. What is enigma? What is enigma? Huh? What is this? Enigma is a special, exceptionally rare and powerful alpha of alphas. Huh? What does that mean? Are we talking about dogs? I don't know. I don't understand. What topic are you studying? I'm studying synthetic biology. Lino. What is menglong? What in the world is that? Menglong? What is that? Who is this? Bruh. It's a Chinese actor. He's dead. Are you saying I'm dead? What the? Bro, I'm not dead. I'm still alive. When is your midterm? Tomorrow. Tomorrow is my midterm. Bima, kamu di mana? I di library. I'm in the library right now. I cannot speak like loudly because I'm in the library. Bima lagi sakit ya. Kemarin sakit. And bibir ay berdarah. Should we go to the norebang? Should we go to norebang? Oh my god. Should I go to norebang? After finishing chapters. Ball 31. There's a sense of single-strand DNA proportionally to time and the nucleus activity. Oh, interesting. It has both exonucleus and nucleus activity. That was DNA P. That was DNA P. And yellow. Nice. Should I memorize all of this actually? I need to pee, bro. Wait, why is it not working? I need to pee. Wait, I'm going to the restroom. I'm back. Study again. Oh, was it not working? It's looking not working. Okay, restriction enzymes as we say. What is the N one? Nucleocapsid protein. I think I should focus more. So I'm going to go now and focus more on studying. Bye bye.